Good morning to all. Today we are going to discuss protein modifications. As you know, the proteins or the nascent peptides that is formed after the process of translation undergoes a lot of modifications before it is utilized at various levels, maybe as an enzyme or a structural component. So there are various applications for the proteins. Before it is used for its intended purpose, such primary translated sequences of these proteins were modified in a variety of fashions, and it is collectively known as protein modifications. As you know, the genome of humans may contain about 25,000 or to 30,000 genes. And from that 25,000 genes, all the required proteins for the function of the human body is created. We know that it is not a simple 25,000 to 30,000 proteins that is available in humans. The complexity of the system is much more. And when we address the complexity of the proteome or proteome complexity, from the 25,000 genes or 25,000 to 30,000 genes, a variety of splicing mechanisms were adopted during the transcription, post-transcription processing or the splicing process to produce maybe more than 1,000 forms of transcripts. As we discussed in previous sessions, maybe that is alternative promoters, alternative splicing or mRNA editing. So various types of post-transcriptional processing of these primary transcripts or the mRNAs were used for creating a diversity of final mRNA products from which corresponding protein can be translated. So the transcriptome size may be more than a lakh, and that is created from the genome size of about 25,000 genes. And this is addressed by a variety of methods during the splicing process, like alternative promoters or alternative splicing or mRNA editing, etc. And again, from the transcriptome, or more than one lakh genes, 1 lakh primary transcripts, 1 lakh transcripts, not primary, processed transcripts of the mRNAs were there, and from them translation occurs. And translation creates proteins, and that represents the proteome. And now you see that how much number? More than a million proteins. More than a million proteins can be created from this transcriptome. More than about 10 times the size of the transcriptome is the proteome. And that is using post-translational modification of the same transcript that is created from the transcriptome. So using different types of post-translational modifications, we can create a variety of new forms of the protein, and that contributes to the proteome diversity. So about a million different types of proteins were created from about 25,000 genes, and that is using post-transcriptional processing and post-translational processing. We already discussed the post-transcriptional methods in previous sessions. Now we will address post-translational modifications which creates such a diverse protein. And the protein modifications may be of different types. There may be amino terminal or carboxy terminal modifications of the proteins. There may be loss of signal sequences, modification of individual amino acids, attachment of carbohydrate side chains to the proteins, addition of isoprenyl groups or prosthetic groups, or even proteolytic processing which breaks the peptide, and formation of disulfide cross-links between peptides to join them together. So all these are various forms of modifications when we discuss. These type of modifications, basically it can be addressed at two levels. Some modifications were treated as co-translational modifications. So all protein modifications were not post-translational. Some of them were occurring during the process of translation itself. So such type of modifications were known as co-translational modifications. Whereas the other ones, after the translation process, is known as a post-translational modifications. We will discuss both the categories. Co-translational modifications were the modifications in which newly synthesized polypeptides undergo co-translational modification, especially in the rough endoplasmic reticulum. The N-linked glycosylation is an essential structural component that is added to the nascent polypeptide in the rough endoplasmic reticulum and is involved in the initial steps of protein folding. So this glycosylation is an essential part that occurs in the rough endoplasmic reticulum, and it is having a significant role in the protein folding process. Again, molecular chaperones. Molecular chaperones were involved in, or they are present in the rough endoplasmic reticulum, and they also facilitate protein folding to generate the correct protein secondary and tertiary structure. So unwanted folding or undesired folding of proteins or the proteins assuming an undesired secondary or tertiary structure is prevented by the molecular chaperones, and they guide these nascent polypeptide to reach its final conformation. So there is a quality control process to ensure that only correctly folded and processed protein exit these rough endoplasmic reticulum. So all these processings which is used to target the protein or target the protein to the endoplasmic reticulum, and the supporting mechanisms like N-glycosylation or the application of the molecular chaperones to help the protein to reach its final correct conformation, all these processes were basically co-translational modifications. Post-translation modifications or PTMs refers to the covalent and enzymatic modifications of the proteins following protein biosynthesis. So it is after the biosynthesis of the protein, complete protein translation. Post-translation modifications increase the functional diversity of the proteome by the covalent addition of functional groups or proteins or proteolytic cleavage to break the chain and create more peptides, or proteolytic cleavage of the regulatory subunits, or degradation of the entire protein and subsequent inactivation of the protein. All these modifications were post-translation modifications. These modifications include phosphorylation, glycosylation, ubiquitination, nitrosylation, methylation, acetylation, lipidation, proteolysis, and influence of almost all aspects of normal cell biology and pathogenesis. Therefore, identifying and understanding the correct post-translation modification is critical in the study of cell biology and disease treatment and prevention. That's why we require a comprehensive study on post-translational modifications. As we listed initially, Amino terminal and carboxy terminal modifications were one among the post-translation modifications. The formyl group, the amino terminal methyl residue, and often additional amino terminal residues, and in some cases even carboxy terminal residues may be removed enzymatically. So the first step of the translation, post-translation modification, is the removal of the formyl group. You know that formyl methionine is present in all prokaryotic proteins. So the first amino acid placed in prokaryotic translation is formyl methionine. So either the formyl group is removed or the entire formyl methionine residue is removed. And in some other cases, if methionine is the first one, like eukaryotic protein, then that methionine can be removed. Again, additional amino terminal residues, if it is required, one or two amino acids additionally, if it is required, that also can be removed. And in some cases, carboxy terminal residues, a few residues from the carboxy terminal end may also be enzymatically removed. So removal of residues, formyl group or some residues from both ends, carboxy or amino terminal ends, that removal is one type of a post-translational modification. In as many as 50% of eukaryotic proteins, the amino group of the amino terminal residues is N-acetylated after translation. So that's another type of a modification. Then there is modification of individual amino acids. The hydroxyl groups of a certain serine, threonine, and tyrosine residues of some proteins are enzymatically phosphorylated by ATP. So that's another process of phosphorylation using ATP. The residues mainly used were serine, threonine, and tyrosine. Then extra carboxyl groups may be added to glutamine residues of some proteins, carboxylation. So that there are examples of some individual amino acid modifications. We will discuss it later, one by one. And another one is a loss of signal sequence. This can be treated as a form of co-translational modification, that a modification that is occurring during the process of a translation. Here you can see an mRNA is represented here. So the mRNA with this 5' cap, it is represented as this one, okay, 5' cap. And starting from this point, the ribosome binds and starts translation, and it moves in this direction. When it is the ribosome, when it reaches the 3' end of the mRNA, normally the translation is nearing its end and the peptide is having more length, and so there will be multiple ribosomes translating the same mRNA, like a polycystonic mRNA. So multiple ribosomes will be translating the same mRNA, and near the end of the 3' end of the mRNA, the ribosome is almost nearing its completion of the translation process with a longer peptide in hand. And here in the cytosol, this is the cytosol part, and this is the endoplasmic reticulum, and endoplasmic lumen is also represented here. The signal peptide and signal sequence, or signal sequence or signal peptide, is the initial part of the peptide that is coded. The 5' terminal of that mRNA is translated into a signal sequence, which may not be required for actual protein function. So the role of this signal sequence, which is attached with every peptide, is targeting that peptide into endoplasmic reticulum. So such proteins which require protein transport to some other locations, usually they need to be packed, processed, and processed properly in the endoplasmic reticulum, folded properly, and even after such modifications needs to be exported to some other cellular locations. So which requires the transportation of this nascent peptide from the cytosol to the lumen of the endoplasmic reticulum. And that targeting and that transport is done using these signal peptide sequence which is initially coded at the starting point of this peptide. So all the peptides, when it is coded, the first segment is the signal sequence. So this is a signal sequence, and there will be another particle known as SRP. SRP is a signal recognition particle. So this signal recognition particle bind to this position, and the signal recognition particle will have a specific receptor on the endoplasm, on the surface of the endoplasmic reticulum. So the signal recognition particle will recognize the signal and during this translation, and it binds to that ribosomal assembly. Then that will be targeted to the signal recognition particle receptor present on the surface of the rough endoplasmic reticulum, where we'll have a ribosome receptor and a SRP receptor. So correctly this will position after binding this SRP, and the SRP receptor will correctly position it, the ribosome, onto the ribosome receptor, and through the channel of this ribosome receptor there will be a peptide translocation complex will be there. So that peptide translocation complex will specifically target this signal sequence of every nascent polypeptide to the lumen of the endoplasmic reticulum. So that will be targeted, and even after, the entire peptide will be transported across this peptide translocation complex into the lumen of this endoplasmic reticulum. So this process is a protein translocation. Now, there is another enzyme which is present here, which is known as signal peptidase. The role of the signal peptidase is to cleave the signal peptide. So once the peptide is targeted into the interior or into the lumen of this endoplasmic reticulum, the role of the signal peptide is over. So that can be removed. It is not required for the final conformation of the protein. So that signal peptide is cleaved and removed, or it is processed and removed by the signal peptidase enzyme, which is present here, immediately associated with that peptide translocation complex. So that will be cleaved and removed. Then the nascent polypeptide now in the rough endoplasmic reticulum is it will be subjected to different type of protein modifications previously, as the ones which is previously described, like binding with the molecular chaperones and getting to the final conformation, N-acetylation, or such different types of additional modifications and chemical modifications which is required to achieve the final structure of that peptide is done within that endoplasmic reticulum. So this is the process of protein targeting to the endoplasmic reticulum, of endoplasmic reticulum, and this is supported by the signal sequence and the signal recognition particle, then which will be directed to the signal recognition particle receptor on the surface of the endoplasmic reticulum. Then the nascent polypeptide will be transported across the membrane of the endoplasmic reticulum through this peptide translocation complex. Signal peptide is cleaved and the protein is released into the interior or into the lumen of the rough endoplasmic reticulum. This is a co-translational processing occurring during the translation in eukaryotic system. So another method of modification is phosphorylation. Reversible protein phosphorylation, principally on serine, threonine and tyrosine residues, is one of the most important and well-studied post-translational modification. So usually this phosphorylation is a reversible process, and that is done for serine, threonine and tyrosine residues. Phosphorylation plays critical roles in regulation of many cellular processes, including cell cycle, growth, apoptosis, and signal transduction pathways. This picture illustrates the sodium-potassium pump. So let's discuss some of the applications of such a phosphorylation process. When we discuss about phosphorylation, it is the attachment of a phosphate group semi protein. Okay, from a high energy molecule, for example, from the phosphate from a ATP can be transferred to a protein. So that's the process of a phosphorylation. So what is the application of this phosphorylation or protein phosphorylation in such biological systems? One best example can be described using sodium potassium pump. Here, you know that there is a sodium potassium pump which is present and across this membrane. For example, this is the intracellular space and this is the extracellular space. And the sodium potassium pump is supposed to expel means from the intracellular space to the extracellular space. They expel the sodium ions through this channel, and it is also supposed to uptake potassium to the interior. So sodium potassium pump will always expel sodium and uptake potassium, and that is the role of the sodium potassium pump. So usually there is a gradient, a concentration gradient. There should be a concentration gradient with a high concentration of sodium outside and a high concentration of potassium inside. So which requires continuous transport of sodium to the outside and continuous transport of potassium to the interior or intracellular space. So this is done by the sodium potassium pump. How it is done? There are specific channels in the cell membrane. So this is the lipid bilayer and the cell membrane. And this channel will have such specific receptors for binding with sodium ions. So this is a sodium channel in which sodium ions will specifically bind here. So once it is bound, this is closed and it cannot go out. But when an ATP molecule, energy-rich ATP molecule come and transfer a phosphate into this channel, the protein which is used in this channel, now it is in an activated state with the phosphate group, then it will have a changed conformation and the channel will open outside. The changed conformation will will expel the sodium ions to the exterior. Similarly, in that changed concentration, another ATP molecule, here for example, here is an ADP and here is this is the phosphate. So once it is bound, it cannot, the potassium ions will bind here, and when the PPI is released, then it is dephosphorylated, the potassium ions will be uptaken. So or it will be intake. This potassium intake will occur when the phosphate is released. Sodium expulsion will occur when phosphate is bound. So this is one of the applications of a protein phosphorylation, a biological application, which can be explained with the sodium potassium pump. So similarly, there are a lot of proteins which will require phosphorylation of the proteins, and the phosphorylation is mainly from such high energy molecules like ATP, and that is one example. And this is another example of a phosphorylation process in a mannose residue. For example, this is a UDP bound N-acetyl glucosamine residue with the UDP. This is the UDP molecule with a N-acetyl glucosamine residue. And here we have an enzyme or a protein with an oligosaccharide mannose residue at one end. So this is a protein with a hydrolyzed enzyme with a mannose residue, which is supposed to or be phosphorylated. So the target of this reaction is to phosphorylate this molecule at the mannose residue. So this is the enzyme with the mannose residue, and the mannose residue will be attached with a phosphate group in this reaction. So for the first step is the expulsion of the UMP. The UDP will be reacting with this mannose residue. So with one phosphate it will be connected with the expulsion of UMP. One phosphate is retained here. Then in the second stage there is a phosphodiesterase and the previous enzyme which is used here is a N-acetyl glucosamine phosphotransferase is the enzyme. And in the second case where we exclude or expel the N-acetyl glucosamine here, this is the N-acetyl glucosamine expelled from the complex. So the enzyme that is used is a diestrase. That is the enzyme that is used. So which will cleave this molecule, or the N-acetyl glucosamine is cleaved, and the phosphate will be retained attached with the mannose residue. So the final product is the phosphorylated mannose residue attached to the protein or the enzyme. So this is one of the examples of a attachment of a phosphate residue to a glycoprotein. Now, so that's about phosphorylation, which we discussed. The next step is about glycosylation. Protein glycosylation is one of the major post-translational modification with significant effects on protein folding, conformation, and distribution, stability, and activity of the protein. Glycosylation encompasses a diverse selection of sugar moiety additions to proteins that ranges from simple monosaccharide modification to nuclear transcription factors, or modification of nuclear transcription factors with attachment of monosaccharides, to highly complex branched polysaccharide changes of cell surface receptors. So there may be a simple monosaccharide addition. Example is a attachment to a nuclear transcription factor, or a highly branched polysaccharide can be attached. Example is a cell surface receptors in some cases. So the glycosylation may be simple or a complex one with monosaccharide, or even a polysaccharide with highly branched complex chain. So different uses of such glycosylation are there. Carbohydrates in the form of asparagine-linked or N-linked or serine or threonine-linked, that means ortho-linked oligosaccharides, are major structural components of many cell surface and secreted proteins. In some glycoproteins, the carbohydrate side chain is attached enzymatically to asparagine residues, which is known as N-linked oligosaccharides, and in others to serine or threonine residues, that is ortho-linked oligosaccharides. Glycosylation of blood cell surface proteins is a good example of protein glycosylation. Here in the lipid bilayer, there will be membrane proteins, and that membrane proteins will be differentially glycosylated using different type of genes which is present in the concerned cells. And the different glycosyl transferase enzymes in different blood groups, ABO blood groups, will add different types of glycosyl groups on the surface of these proteins. Based on that glycosylation or differences in the glycosylation, we will have A, B, or O blood groups that is created. So that is one of the examples of the role of glycosylation of the membrane proteins in blood cells in determining the blood groups. Now, this is another diagram that shows the glycosylation process. Here we have N-acetyl glucosamine, mannose or glucose, various types of sugar residues are here, and that will be gradually added, and this chain is developed by combining all these types of sugar residues. The chain is created here. Various enzymes will interact in adding the chain or residues to the chain, or by removal of the residues from the chain. And finally, such branched polysaccharides will be added to the asparagine residues in the nascent polypeptide that is created. So now it becomes the glycoprotein. So this type of glycosylation of proteins is one of the post-translational modification that is present. Here we have N-glycosylation, that is addition of these glycosyl groups to the NH2 residues of the protein. O-glycosylation to the OH groups, glycosyl groups added to the OH groups, glycation, that is attachment of these sugar residues to the COH group. Then C-glycosylation is another one. Then glycosylation is another one. So there are different types of glycosylation methods based on where these the glycosyl groups were attached. So there are different types of glycosylation process. Basically, most prominent ones were N-glycosylation or glycosylation. S-nitrosylation is another protein modification. Nitric oxide, in short NO, is produced by 3 isoforms of nitric oxide synthase. And it is a chemical messenger that reacts with the free cysteine residues to form S-nitrothiols or SNOs. S-nitrosylation is a critical post-translation modification used by the cells to stabilize proteins, regulate gene expression, and provide nitric oxide donors. And the generation, localization, activation, and catabolism of these nitric oxide synthase are tightly regulated. S-nitrosylation is a reversible reaction, and SNOs have a short half-life in cytoplasm because of the reducing enzymes including glutathione and thioredoxin that denitrosylate these proteins. So these two enzymes, glutathione and thioredoxin, which is present in the cytoplasm, will denitrosylate these proteins. So nitrosylated proteins will be denitrosylated in the cytoplasm. So we can say that this is a reversible reaction. Therefore, SNOs are often stored in the membrane or membrane vesicles and the interstitial space of the lipophilic protein folds to protect them from denitrosylation. Usually, if it is present in the cytoplasm, it will be denitrosylated and the reaction is reversible. So, usually such type of nitrosylated proteins will be kept within some membranes, vesicles, or some interstitial space, or within some lipophilic protein folds, etc., to protect it from such a enzyme activities which is occurring in the cytoplasm. For example, caspases, which mediate apoptosis, are stored in the mitochondrial intermembrane space of the SNOs. Spaces are SNOs. And in response to extra or intracellular cues, the caspases are released into the cytoplasm. And the highly reducing environment rapidly denitrosylates the proteins, resulting in caspase activation and the induction of apoptosis. So this is one example of such denitrosylation of caspases in the cytoplasm. And that may result in the apoptosis. So this is some of the examples of modified amino acid residues. Phosphate attached with the serine, phosphoserine, or phosphothreonine, phosphotyrosine. Phosphate group is attached. Then carboxyglutamate, for example, carboxyl group is attached. Then methyl lysine, dimethyl lysine, etc., where methyl group is attached, and trimethyl lysine, methyl glutamate, etc., where methylation occurs. So phosphorylation, carboxylation, methylation, etc., are some of the post-translation modifications or specific amino acid modifications. And methylation can be discussed in detail. The transfer of one carbon methyl groups to the nitrogen or oxygen, that is N or O methylation. So wherever it is attached. So if it is attached to a nitrogen, that is N-methylation. If it is to the oxygen, that is O-methylation. And this type of a transfer of one carbon methyl groups to carbon or nitrogen of the amino acid side chains increases the hydrophobicity of the protein and can neutralize the negative amino acid charge when bound to carboxylic acids. Methylation is mediated by the enzyme methyl transferases. So S-adenosyl methionine, that is a SAM, is the primary methyl group donor. N-acetylation, or the transfer of an acetyl group to nitrogen, occurs in almost all eukaryotic proteins through both irreversible and reversible mechanisms. The N-terminal acetylation requires the cleavage of the N-terminal methionine by methionine aminopeptidase or MAP before replacing this amino acid with the acetyl group from the acetyl coenzyme A by N-acetyl transferase or NAT enzymes. This type of acetylation is co-translational in that N-terminus is acetylated on growing polypeptide chains that are still attached to the ribosome. So N-terminal methylation, N-acetylation. Methylation is N-acetylation is a co-translational process because it is occurring. This N-acetylation occurs while the peptide is synthesized, or so it is attached to the growing polypeptide chain. The ribosome is already attached with the chain, so it is a co-translation process. While 80 to 90% of the eukaryotic proteins are acetylated in this manner, the exact biological significance is still unclear. This is about the histone modification. We can say that this is an application of acetylation and methylation process. You know that the histone proteins, usually the DNA is wound over the histone proteins, and that is used for the condensation of the chromatin to the heterochromatin structure. So heterochromatin is the condensed form, euchromatin is the relaxed form. So when methyl group is attached, what happens is that the heterochromatin is formed, that is with the methylation, the histone proteins will favor the condensation of the chromosomes with the chromosome winding over that histone proteins. Whereas if it is acetylated, what happens? Then the chromosome, there will be a change in the structure, and the histone proteins will unwind the DNA which is wound over that one, and a more relaxed euchromatin is produced when it is acetylated. So methylation and acetylation, these processes is having a significant role in even the gene regulation and gene expression of the eukaryotic system, because the chromosome packing and unpacking, the condensed and relaxed structures, heterochromatin and euchromatin structures of the eukaryotic genes is having a significant role in regulating the eukaryotic transcription process. Because if it is wound tightly over the histones, and if it is present in the condensed form, then that will not be available for transcription. So it is the gene is in an inactive state. So methylated histone proteins means gene is not active. And if it is acetylated, then that means the DNA is unwound. It is present in the euchromatin state. That means the DNA is available for transcription machinery to transcribe and produce the protein. So gene is in an active state. And this slide shows some post-transcriptional modifications associated with the histone proteins. So there are acetylation, methylation, phosphorylation, and ubiquitination of various histone proteins: H2A, H2AX, H2B, H4, and H3. So different histone proteins can be methylated, acetylated, phosphorylated, or ubiquitinated at various points. So this diagram shows the overall locations where such chemical modifications can occur. Lipidation is another modification. Lipidation is a method to target proteins to membranous organelles, endoplasmic reticulum, Golgi apparatus, vesicles, including the endosomes and lysosomes, and the plasma membrane. The four types of lipidations listed here are C-terminal glycosyl phosphatidyl inositol, or GPI anchor, then N-terminal myristoylation, S meristylation, and S prenylation. These are four types of lipidation processes that we will address. Each type of modification gives the proteins distinct membrane abilities. Although all types of lipidation increases the hydrophobicity of the protein and thus its affinity to the membranes. The different types of lipidation are also not mutually exclusive because in that two or more lipids can be attached on the same protein. So it doesn't mean that a single type of lipidation occurs for a single protein. Maybe in the same protein, different types of lipidation can simultaneously occur. This is the first category where we discuss the GPI anchors, and GPI anchor is used for attaching such proteins to the plasma membrane, where we will have, this is the lipid bilayer membrane, and this is the GPI or GPI anchor, which will have a lipid component, and lipid component will be incorporated or into the lipid bilayer or the membrane. And this lipid anchor, and the second component of the anchor is a glycan protein, or a glycan, that is a what is called a polysaccharide component. Then the protein is attached. So these GPI anchors were used for attachment of such proteins to the membranes using such GPI anchors. And GPI anchor will have glycan and lipid components. The lipid component is used for embedding this structure into the lipid bilayer of the plasma membrane. GPI anchors tether cell surface proteins to the plasma membrane. And these hydrophobic moieties are prepared in endoplasmic reticulum, where they are then added to the nascent polypeptide en bloc. GPI anchored proteins are often localized to cholesterol and sphingolipid-rich lipid rafts, which act as a signaling platforms on the plasma membrane. And this type of modification is reversible, as this GPI anchor can be released from the protein by phosphoinositol specific phospholipase C. Indeed, this lipase is used in the detection of GPI anchored proteins to release GPI anchored proteins from the membrane for gel separation and analysis by mass spectrometry. So this is having various applications even in the detection of GPI anchored proteins. Now, the next category of lipidation is N-meristylation. N-meristylation is a method to give proteins a hydrophobic handle for membrane localization. This meristoyl group is a 14-carbon saturated fatty acid, C14, which gives these proteins sufficient hydrophobicity and affinity for the membranes, but not enough to permanently get anchored into the protein or anchor the protein into the membrane. So it is having hydrophobicity and affinity for the membrane, but it is not permanently attached or anchored to the membrane. N-meristylation can therefore act as a conformational localization switch in which protein conformational changes influence the availability of the handle for membrane attachment. Because of this conditional localization, signal proteins that selectively localize to the membrane, such as Src family kinases, are N-meristylated. The third category is palmitylation, in which it adds a C16 palmitoyl group from palmitoyl coenzyme A to the thiolate side chain of the cysteine residues via palmitoyl acetyl transferase. So in the previous case, it was a C16 group addition. This is a C14 group addition. This is a C16 group addition. Because of the longer hydrophobic group, this anchor can permanently anchor the protein into the membrane. So the overall purpose of such lipidation process is anchoring such proteins into the lipid membranes or to the cell surface. The localization can be reversed or it can be released, though, by thioesterases that break the link between the protein and the anchor. Thus, S-palmitoylation can be used as an on-off switch to regulate the membrane localization. S-palmitoylation is often used to strengthen other types of lipidation, such as methylation or pharyngylation, and S-palmitoylated proteins also selectively concentrated lipid rats. So this is another type of lipidation in which the protein is strongly anchored using its longer hydrophobic loops into the membrane. The next category is prenylation. It's another pharyngeal group or a C16 group addition, and or there is a geranyl group, which is a carbon 20 group to specific cysteine residues within the 5 amino acids of the C terminus via pharyngeal transferase or geranyl transferase enzymes. So that type of anchor is used for protein anchoring. Unlike S-palmitoylation and S-prenylation, this is hydrolytically stable. The previous ones were reversible. This is hydrolytically stable. Approximately 2% of all proteins are prenylated, including all membranes of the RAS superfamily. This group of molecular switches is that is RAS family molecular switches is pharyncylated, geranylated, or a combination of both is used because it is a more stable incorporation. Additionally, these proteins have a specific 4 amino acid motifs at the C terminus that determine the type of prenylation at single or dual cysteines. So the next topic is about protein degradation. So there are protein modifications, and even protein degradation is a part of protein modification, and we will discuss that one also. And the main modification for protein degradation is ubiquitination. And the ubiquitin is an 8 kDa polypeptide consisting of 76 amino acids that is appended to the NH2 of the lysine, and lysine in the target proteins, and via C-terminal glycine of ubiquitin. So the C-terminal of the ubiquitin is attached to the NH2 of the lysine. The ubiquitination is also known as the kiss of death process for a protein. In ubiquitination, a protein is inactivated by attaching to ubiquitin to it. So usually the protein is active, it is inactivated by attaching ubiquitin, and the ubiquitin acts as a tag that signals the protein transport machinery to ferry or transport this protein to the proteasome for a degradation. So there will be a proteasome which normally degrades these proteins and unused proteins. So ubiquitin, when it is attached to such proteins to be degraded, then it will act as a signal or tag for protein which is intended for degradation. So that will be the protein transport machinery will transport all the ubiquitin target proteins to the proteasome for degradation. Following an initial monoubiquitination event, the formation of a ubiquitin polymer may occur. So usually it is initially one ubiquitin group is added, and later multiple ubiquitin may be added, and a polymer of ubiquitin is attached to the protein. And polyubiquitinated proteins are then recognized by 26S proteasome that catalyzes the degradation of the ubiquitinated proteins and recycling of the ubiquitin. So once the protein is degraded, the ubiquitin is again recycled for degradation of another protein intended for degradation. So this is the overall structure in which we have a target protein. So the target protein is marked in pink over here. This is a target protein. So some recruiter molecules initially that recruits this protein, and then an assembly of the proteins which will guide these protein, and then finally ubiquitin molecules were a single molecule. This is a monomer, and once multiple monomers were attached, then a ubiquitin polymer is attached to the protein. So this will act as a label or a tag for protein to be degraded. Once the ubiquitin molecules were attached to the protein by this machinery, then this protein is a target for a degradation. Then proteasome, 26 S or sedimentation coefficient, that is the capacity, the size of this proteasome. that will, or the protein transport machinery, once it is ubiquitinated, the protein transport machinery in the cell will transport this to the proteasome, and then it will be degraded, and once the protein is degraded, these ubiquitin molecules will be available for reuse to be recruited later. So this is another representation of the same, in which we have the target protein, which is represented at this point. The target protein will bind with E2 and E3. So once then here we have the ubiquitin conjugation. Ubiquitin molecules is represented as around here. So the E2 will bind with the ubiquitin, and that will be attached to the protein. So the protein that is intended for degradation will be attached with the multiple ubiquitins, and that is the ubiquitin polymer. So once such a polyubiquitinated protein is identified, then protein transport machinery will transport it into a 26S proteasome for degradation, and the protein will be degraded, and polyubiquitin will be further deubiquitinated to form the ubiquitin monomers, which will be recruited by the E1 and transported to E2, and the E2 will again attach to the next protein target for degradation. So such polymer, this cycle continues, and that's how protein degradation occurs. So the next topic is about proteolysis. That is the last one, and proteolysis is the degradation is also another form of degradation of the protein, and peptide bonds are indefinitely stable under physiological conditions normally, but and therefore cells require some mechanism to break down these bonds. Usually at physiological conditions, the peptide bonds are stronger, that will be stable for longer periods. But protein degradation requires, or proteolysis requires specific mechanisms for breaking the bonds of the peptides. The proteases comprises a family of enzymes that cleave these peptide bonds of the proteins and are critical in the antigen processing, apoptosis, and surface protein shredding and cell signaling. Wherever such protein degradation or protein cleavage is required, we require proteases. The family of over 11,000 proteases varies in substrate specificity, mechanism of peptide cleavage, location of the cell and where it acts, and the length of the activity. So based on all these things, there are about more than 11,000 proteases are there. Okay. Proteolysis is a thermodynamically favorable and reversible reaction. So once it is cleaved, then it cannot automatically rejoin. So it's a thermodynamically favorable, irreversible reaction. Therefore, protease activity is tightly regulated to avoid uncontrolled proteolysis through temporal or spatial control mechanisms, including regulation by cleavage and compartmentalization. So if a protein is to be protected, that needs to be such type of proteases. It cannot be left uncontrolled in the cytoplasm where the remaining proteins were there. So proteases were usually confined within specific compartments like proteasomes or lysosomes within which they act. So it cannot be left unregulated in the cytoplasm, because it will have unintentional cleavage effects on target proteins. And of course, it is since it is an irreversible reaction, it will do much more damage if it is unregulated. So usually such proteases were compartmentalized, where like proteasomes or lysosomes, where its activity of the protease is specifically confined to that protease, to that compartment. Okay. Based on the site of action, we have two categories, that is aminopeptidases and carboxyl peptidases, where these acts, the site of action, that is used for cleaving this bond, because aminopeptidase is one category and carboxyl peptidase is another category, from which end it is cleaved. And the next one is about based on the active site or the groups, active site groups that is used for cleavage. So there are serine protease, cysteine protease, aspartic acid protease, zinc metalloprotease. So which active site it recognizes for cleavage. So that based on which we have different types of proteases. So proteolysis, actually it is two types of catalytic mechanisms were reported. One is acid-based method, or in which the acid group of the enzyme, that is like aspartate, glutamate, or metal ion, which are acidic compounds. So that acid group of the enzyme will attack or will perform a hydrolytic action of the peptide. So the acid group will hydrolyze the bond, peptide bond, and breaking the chain. So you will have two products. So here is a hydrolytic action of the acid group of the enzyme. And some examples were aspartate or glutamate or metal ions, etc., will have such acidic groups that will result in hydrolytic action and cleavage of the proteins. The second type is a nucleophilic attack. Some proteins, like some enzymes, which will have cysteines or serine or threonine, which may perform a nucleophilic attack on the substrate and then thus cleave that enzyme. So there are maybe two types of mechanisms of proteolysis. One may be acid hydrolysis and another one a nucleophilic attack. In both cases, the peptide bond will be broken, and you will have two products, or it will be fragmented. And that is the mechanism, two types of or two methods of proteolytic cleavage. So this is about the different types of post-translational and co-translational modifications. In short, we can say that they are protein modifications. Thank you.